Hello, this is Dr. Ann Iona Avril, Associate Professor from the Department of Zoology, Holy Cross College, Tiruchirappalli. I am going to deal a topic on locomotion in protozoa, pseudopodia, flagella and ciliary movement. Upon learning this topic, the learner will understand the types of protozoan locomotion. Describe the mechanism of pseudopodial locomotion. Understand the flagellar movement in protozoa. Describe the ciliary locomotion in protozoa. Protozoa, they are microscopic, single-celled organism that inhabits a wide range of aquatic and terrestrial environment. Despite their small size, protozoa exhibit a wide variety of locomotory strategies. That is what I told you about pseudopodia, flagella and ciliary movement. So there is a wide range of locomotory strategies. This enables them not only to move through their habitats in search of food, avoid predators and find suitable environment for reproduction. There are three primary modes of locomotion which is observed in protozoa: pseudopodia, flagella and ciliary movement. We will see about pseudopodia. Pseudopodia in Greek, it means false feet. They are temporary and cytoplasmic extension that some protozoa, not all protozoa, some protozoa use for movement. This mode of locomotion is a particular characteristics of an amoeboid protozoa. The ability to form pseudopodia is a defining feature of this group, and it is made possible by the dynamic properties within the cytoplasm. So cytoplasm is going to play the major role in outpushing an extension which we call it as pseudopodia or the false feet. Look at this picture where you find this is the amoeba, and the amoeba through its cytoplasm, it gives an outpushing, an extension which is very flexible, and this there are many which is used for trapping the food as well as for moving forward. They use as for locomotion. So all these are the pseudopodia shown in the picture. Structure and formation of pseudopodia. Pseudopodia, they are not rigid structures, but they are flexible and gel-like extensions of the cell membrane. They can take various shapes ranging from small finger-like projection to a broad lobe depending on the specific protozoan, the specific species of protozoa and its environmental condition. The key to pseudopodial movement lies in the underlying cytoskeleton. The key is actually the underlying cytoskeleton, and that cytoskeleton is made up of a protein called actin, and that actin gives rise to filaments. The formation of pseudopodia begins with the polymerization of the protein called actin monomers into filaments at the leading edge of the cell. Wherever the cell has to move forward, at that tip you find the actin gets polymerized. This creates a meshwork of actin filaments which pushes the membrane outward. Concurrently, myosin, another motor protein which is meant for movement, it interacts with the actin filament to generate a force called contractile forces, allowing the protozoan to pull its trailing end forward. As a result, the cell undergoes a continuous cycle of protrusion, adhesion and retraction, enabling it to move in the direction of its pseudopodia. Look at this picture now, how an amoeba with the outpushing or extension called pseudopodia, to start with there is a protrusion and then slowly it gets attached. It gets attached to the substratum, and after it gets attached to the substratum, then it releases again. So there is a protrusion attachment and retraction. Look at this dark object here in the substratum, that is the food particle. With the help of this pseudopodia, in the next picture, you will see that it engulfs the food particle, making use of the process known as phagocytosis, engulfing the solid particle. Functions and adaptations about this pseudopodia. Pseudopodial movement is highly adaptable and responsive to changes in the environment. So this is an adaptation to the protozoa having the pseudopodial movement. Protozoa with pseudopodia can move through a variety of substrates from muds and sand to organic debris and even inside other organisms. They can also engulf food particles through a process called phagocytosis. Using their pseudopodia, it surrounds and engulfs the prey or the detritus. Next, we will see about flagella. Flagella, they are very long, lengthy, and they are whip-like appendages that protrude through the surface of the certain protozoa, enabling that protozoan to swim or propel themselves through liquids. Flagellated protozoa include groups like kinetoplastids, diplomonads, and various types of green algae. Structure and function of the flagella. Flagella consists of a microtubule. Unlike the pseudopodia has actin and myosin, you find here the microtubule-based structure called axoneme, a new terminology being introduced in flagella, which is enclosed within the cell membrane. Now, this axoneme is composed of a characteristic called 9+2 arrangement. This microtubule 9+2 arrangement, which means they are with 9 outer doublet microtubules surrounding 2 central singlet microtubules. 9 outer doublet and singlet with 2 central microtubules. A protein known as dynein motor proteins. They are located on the microtubules, generates the force required for the flagellar movement. This picture shows the protozoan having a whip-like structure. That whip-like structure is the flagella, which brings about the movement. Flagella beats in a coordinated whip-like motion, which propels the protozoan through its liquid environment. This movement is typically characterized by a power stroke, during which the flagellum is actively bent, and a recovery stroke, during which it returns to its original initial position. The precise coordination of these strokes allows the protozoan for an efficient forward movement. Now, this picture gives you a clear idea about how there are different stages of flagellar movement. Look at this picture, Euglena, for taking an example of Euglena. At the base of the stigma, you find a whip-like structure called flagella. So, the flagella has a lateral force. When it has a lateral force, it bends down, and similarly, after some time, it has a longitudinal force moving forward, and then it comes back to the original position. So, this picture shows one Euglena undergoing different force, the lateral force, longitudinal force, and forward thrust for the flagella to move. Stages of a euglenoid movement, there are different stages, 1, 2, 3, 4. Similarly, 1, 2, 3, 4 forward. These are when the contraction starts, the euglena moves. So with that, the flagellary movement is over. The next one is the ciliary movement. Cilia, they are short and air-like structures that are similar in structure to that of flagella, but they are typically, cilia are smaller, shorter, and numerous in number. They are found in the cell surface. Cilia are found in various protozoan groups, including ciliates and some flagellates. They are also present in many other eukaryotic organisms, including animals. This picture shows the difference between a cilia and a flagella. Both are protozoan. Now look at this protozoan, which has numerous short air-like structures called as cilia. Now this is another protozoan having very lengthy whip-like structure and few in number. They are flagella. So structure and function of cilia. Cilia shares the same microtubule based structure 9+2 arrangement as flagella with dynein motor proteins driving the movement. However, cilia are shorter than flagella and are often present in large numbers, covering the entire cell surface or a specific regions. Ciliary movement is characterized by coordinated, rapid and synchronized beating of the cilia. I like to stop here and take you to a green field, a paddy field. When you cross the paddy field, you will look at the motion of that paddy leaf, how according to the breeze they will show a rhythmic movement. You call that as a metachromal rhythmic movement. Similarly, the flagella also shows a movement, a wave-like movement. The motion creates currents in the surrounding fluid, which can be used for various purposes. In protozoa, cilia primarily serve for locomotion, helping to propel the cell through aquatic environment. And this is what I told you, how there will be a direction of the cilia, how they move. So the entire body is covered by means of a short air-like structure throughout the entire cell surface. Taking one particular portion, I could show you in this diagram how the cilia moves. So this moves in a circular movement, almost like a wave-like action when you see together. To conclude, protozoans are a diverse group of microscopic organisms that have evolved a wide range of locomotion strategies to navigate the environment effectively. Pseudopodia, flagella and cilia represent the fundamental mechanisms that enable the protozoan to move, feed and interact with the surroundings. The locomotion strategies are not only essential for survival and reproduction, but also play a crucial role in shaping the ecosystem and serving as an indicator of environmental health. The adaptability and diversity of protozoan locomotion highlights the remarkable activity of these small, often overlooked organisms. Further research in protozoan locomotion promises to unveil more about their biology, ecology and potential. These are the frequently asked questions, and these are the references used for this particular topic. And the protozoa, though it is small microscopic, it has a wide range of locomotory activities, making use of the three different types, namely Pseudopodia, cilia and flagella. Thank you.